Welcome to today's lecture, which is the last lecture in the infectious diseases and inflammation seminar series in the postgraduate lecture course. So I asked Graham Brown to give today's sort of closing lecture on global health, or the impact of infectious diseases globally, and also the initiatives that are being done. Where research has led us and where it still needs to lead us in terms of policy changes. And Graham is perfectly placed to give today's lecture because he's really been at the forefront of all of those aspects of research and global health. And he's mentored many people, including myself on a personal level. Graham's responsible for my infectious diseases training, but also responsible for getting me involved in infectious diseases research. So Graham actually still has been involved with the WHO for a long time, and he still is the chair of the Malaria Vaccine Strategic Advisory Committee with the WHO, and he's also the foundation director of the Nossal Institute. He was formerly the head of the Infection and Immunity Division here at WEHI, then became the director of the Victorian Infectious Diseases Service at the Royal Melbourne Hospital, professor of medicine at the Royal Melbourne Hospital, before moving on to become director of the Nossal. So it's a great pleasure to have you.
These former students sometimes feel they owe you something. Is that microphone on?
Yes.
Okay. I was just reminiscing to Mark, when I came here as a PhD student, you would have thought that the immune system was designed to work out the immune response to DNP coupled to ovalbumin in a bone marrow, thymectomized, wholly aerated, transplanted Miller-in mice. And it took, occasionally you forgot that actually the immune system's got something to do with infections and the body's response. So as someone who's interested in infectious diseases, I wanted to do a couple of things today. One is to try and put the context of infectious diseases into that balance that it always exists, and I'm sure others in this series have spoken of it, that the outcomes are a balance between the microbe, the epidemiology, or the environment, if you like, and the host. And I think malaria is a wonderful example of that this balanced relationship that sometimes leads to disease, or immunopathology causes disease, is all part of a good example of how infectious diseases, immunity, and the global picture come together. So what I want to tell you a little bit about is to remind you of that theme, what infectious disease is. Then I want to talk about malaria, the disease, as opposed to malaria, the infection, and the various consequences. And then to the bigger picture, if we're looking at a global approach to prevention of malaria, what are the tools that we have? And then Bill and Melinda Gates called for eradication of malaria. Is it reasonable? Is it possible? And why did they do it? And then what's been the global response and the challenges? So when we think about infectious diseases, it really is the outcome of that disturbed balance between the host, the parasite, or in this case the parasite, host pathogen, and the environment. And it's very useful often with infectious diseases to think what's in it for the organism. An organism that kills its host rapidly before transmitting is not very effective as an organism for living a long time. So we see this particularly in parasitic diseases, where you have a balanced relationship of chronic infection that's not causing much harm to the host is ideal for survival of that organism, such as the bacteria or viruses that we carry all the time. That relationship is incredibly important, and it's when that relationship becomes disturbed that we have disease. And I think it's really important to recognise that the organisms only exist and survive because they have, in our case, we're looking with human hosts, sometimes get tangled up with animals as well, but mainly humans. In fact, most infectious diseases of human origin came from animals originally. And I think it's very helpful to consider interactions of populations of organisms and populations of people. If the asylum seekers get malaria in Manus Island, what's the cause of malaria? Australian government policy, the weather conditions of Manus, or the mosquito. The point being that you require a combination to have that final outcome. And similarly, from a public health perspective, preventing those outcomes, you can do it in various different ways. Also, you'll hear emerging infectious diseases. I can imagine this series, I think someone spoke on flu, And so we think about a newly appeared organism, or one that's been out of sight for a long time and then comes back. Maybe re-emerging, such as emergence of resistant tuberculosis or malaria, or emergence of resistance to HIV as some sorts of examples. So these are ones that have emerged recently, and in each case one can imagine that these are disturbance of that host, pathogen and environment. So Ebola coming from animals in West Africa, probably people eating bush meat become contaminated and infected by this virus with lethal consequences. You know the flu story coming from chickens. Of course, flu is really an avian virus, and we are by chance infected in that regard. Alternatively, we know Creutzfeldt-Jakob disease and its new variant, bovine spongiform encephalopathy, changing of rendering of material. So sick animal products were churned up and fed back to cattle, and that's how it came. And similarly, in the great diseases that are of terrible importance to developing countries, malaria, HIV and TB, drug resistance is a problem in all of them. And so just to make that point, and I'm not going to take this theme a lot longer, but some examples that I've just described there is to suggest how these new diseases emerge as a disturbance in that balance. And sometimes we don't really know where they started, like SARS, which came out of the blue and caused a certain number of deaths some years ago. We don't really know where it started. Perhaps it's carried by bats, who knows, and it may emerge again, of course. And we also know that different flu variants can also arise. So speaking of, back to that theme, of course the vector for malaria is the Anopheles mosquito, and you'll see that this mosquito is recently fed, had a big blood meal, and it's a very powerful drive to feed. Prior to laying eggs, a female mosquito must have a blood meal, and that's why if you're in a tent and they'll keep buzzing and buzzing and buzzing until they find you to get the blood, that is a very, very powerful drive. And of course within the mosquito there is a sexual form of transmission. It's just not like a needle exchange with someone getting hepatitis transmitted from one to other, the fact that there happens to be a bit of blood carried, but more there's actually part of the sexual cycle and recombination which occurs within the mosquito. So when we're thinking about this host, pathogen and environment, mosquitoes only got a certain lifespan, of course, and if the mosquito probably feeds only every 2 days, so if a mosquito feeds for its first blood meal on a person with malaria, then it will take more than 2 days or 4 days until it's infectious. So at the next meal, 2 days later, they will not be infectious. The next they may be, and subsequently. So if you could reduce the life of mosquitoes just by a day or two in some magic way, this would be a very, very important way of reducing malaria. And when you model, you have to bring in what's the life of a mosquito and why to understand about transmission. Because when we come at the end of the talk to about how we get rid of malaria and eradicate it, the absolutely key thing is this R0, or the reproductive number, which for any infectious organism relates to the number of secondary cases you get from a primary case. So if you could shorten the life of every mosquito, you'd have an enormous effect. So these are details, but it's just saying how complex this interaction is. Similarly, if the weather changes in Manus Island, or the weather changes where these mosquitoes are breeding, this can not only affect the life, but the length of life, but also affect their flight range and many other things that are so important with respect to transmission. Now, the other thing which will be important is just a little bit about the life cycle. The forms that the mosquito injects into you first go to the liver for 10 days or so, and they don't cause any symptoms at all. Symptoms occur when these parasites come into the bloodstream, and then they circulate, and this is what makes you sick. The immunopathology of malaria from the blood forms is what makes you sick. Every once in a while, and triggers are not well understood, but sexual forms are produced, and these are the ones taken up in the mosquito. Now, that's the story for Plasmodium falciparum, which is the most severe one and the main one you'd hear about in lectures here. But another form called Plasmodium vivax, it does exactly all that, but it also has the characteristic that it can sit in the liver to emerge months or years later, so that the person can be absolutely asymptomatic and then have malaria 3 years later, for example. And it's absolutely amazing biological adaptation, because this is in fact how malaria survives the Russian winter. That form of malaria survives very well. What happens is the parasites remain dormant, and in the cycle, which varies depending where you are in the world on when it comes back, at springtime, who knows the stimulus, but what happens is that the parasites emerge and the cycle goes on. So Plasmodium vivax, which is the secondary form and will be very important when we talk to elimination of malaria, is able to persist in this way for a very, very long time. So you don't need a very intense, constant transmission because of this ability. So you can have an oasis in the desert where you have Plasmodium vivax without a big population, whereas with falciparum you need quite a large population. We'll come back to that in a minute because it's particularly interesting. So it's very important to know when you're talking of malaria, it means different things to different people, so I'll just show you in a moment. So it's infection or it's disease. If you're trying to stop people being sick and dying, you're most interested in the disease aspect and stopping kids dying or pregnant women from getting seriously sick. But if you're interested in getting rid of it for all time, you've got to be aware of infection, and the person may be totally asymptomatic. There are types that cause lethal diseases, Plasmodium falciparum. There are very many different antigenic strains of this, and the individual parasites themselves can undergo clonal antigenic variation, and you can start with a single parasite and then show that it changes from A to B to C to D. In fact, that work was done here in our lab with Bev Biggs to show that individual parasites can change. So this isn't about diversity, it's about clonal generation of variants, which is extremely interesting. And interestingly, about those variants, the antigens that they express on the surface of infected cells are different. So they're antigenically distinct, but they're also phenotypically distinct, to the extent that certain ones can bind in some organs and not in other organs. Lots of organisms alter the antigens they express with diversity, but it's not all that common for them to have different phenotype in association. It does have a little bit, for example, in influenza, if you've no doubt heard, as to whether influenza is able to bind high or low in the respiratory tract, the sort of dose that's required to get into a place where those viruses can bind, but it's a very interesting aspect of falciparum. So, and as we'll see in a moment, although malaria can cause this severe problem of cerebral malaria, it can also cause severe anemia very commonly. And it's extraordinarily interesting in parts of Africa where there's very, very intense transmission of malaria. All babies would get malaria within the first few months of life. So they get over a bit of their maternally acquired immunity. And then where there's a lot of malaria, the peak problem with malaria is at 1 year with severe anemia. In places where malaria is intense, but not quite so intense, the main problem is in children a bit older, about 3 years of age, and they get cerebral malaria. Now, there are not many diseases where usually the first contact with the disease gives you the worst dose of it. It's a little bit unusual, therefore, to have multiple episodes of infection and then get different manifestations at different times. Not very well understood at all. Dengue is one example where you can have more serious disease at your second infection, so-called immune enhancement. But we don't really understand why that's the situation with malaria. I've mentioned the problem with Vivax relapse and the fact that at the moment, I'm sure you've had talks here about vaccines and that works. The first prototype vaccines are quite well advanced, and there might be by 2015 a partially effective licensed vaccine. And as we've said, babies, children, pregnant women are the ones at greatest risk from this disease. So this is a child in Madang, Papua New Guinea, where many of us have worked. So here's a young child having crying, hot, febrile. You don't know what it is until you do a blood test, and that's done with a finger prick and look under a microscope. And then what you see in the diagnosis on a thin film is you see these little spots, which are malaria, technically hard to diagnose, and sometimes with poor microscope and poor stains, a real problem. And many of the places where malaria is diagnosed, they don't have electricity, let alone a microscope or a person who can read those slides. And so there's been a huge effort to try and develop rapid diagnostic tests, antigen detection tests, which have made a great difference and are not bad for Plasmodium falciparum, but that's the serious form, but are not very good for Plasmodium vivax, unfortunately. The other problem, as you'll see in a tick, is that children can be immune to malaria yet have parasites in their blood. This is a very common feature of parasitic disease, that you can have infection with immunity. I give the example for people who are not in the tropics, say a cold sore, where you could have a cold sore. What, a third of us, whatever the number in this room, have had cold sores at some stage in our life, but at the moment you can't see anything, but you know that it's dormant there somewhere. Then it can flare up and you'll see it for a while and it'll disappear. The same thing is true with malaria. This concept of concomitant immunity is really interesting and very important. And in fact, it's so important for malaria that you need constant boosting with infection to retain that state of immunity. The sort of work that's been done is taking people who are African students, immune to malaria, and then what happens? They go to Europe, study for a few years without having any exposure to malaria, they go back home and they're at risk again. So it's a very interesting aspect of this disease, once again, not very well understood, but I'll come back to it to emphasise it. So this is the form we're talking about, Plasmodium falciparum malaria. This is Plasmodium vivax, the one that's relapsing. These are other less common forms, but if you're eradicating malaria, you've got to get rid of these as well. These are the asexual forms of these parasites, which don't cause any symptoms, but an asymptomatic person therefore can be a very important reservoir of ongoing infection. By contrast with the child who just had a febrile illness, here's a child who suffered the serious consequence of cerebral malaria. And you'll see this is not a baby, it's a child a few years old who would have had many episodes of malaria and then got this severe form. Why is it so? Did he develop some IgM or something that causes the problem in the brain? Is it a different strain, different species? Is it immunopathology from cytokines? Still not understood. Probably the precipitating event is that these malaria-infected cells, this is a section of brain, relatively normal brain here, and a capillary, and all these black dots are malaria-infected red cells blocking this capillary. It's clear that this is an important aspect of cerebral malaria in humans, but it's not the only thing. There are huge cytokine responses as well, which are very important. Sometimes you see changes in the brain as well. And it's really interesting, those who believe a sort of mechanical sequestration hypothesis, they say, well, it's a bit like a stroke. Well, if you had a stroke with such profound blockage, it would take a long time to recover. But strangely, with malaria, a child can be unconscious with cerebral malaria and within 24 hours up and running around again. Clearly, there's a combination of factors that contribute to this severe disease. Funny thing, therefore, a disease that can come after multiple episodes. We don't really understand the pathology. It's partly to do with sequestration, but other things involved. And I'd always like the immunologist in the room to think about why should that be so? It's so unusual for an infectious disease. This is obviously modelled, and a lot of work's been going on in this institution in understanding the mechanism by which these infected red cells, you can see the changes on the red cell sticking to cells here for laboratory study. Now, having what I've said then is if the children get through these first few years of life, some die of the cohort, but most survive, and then these children have an immunity that protects them for the rest of their life, provided they have constant challenge. So what's really unusual is that in getting to that stage, you can have protection right through to immunopathology that we've seen before. And interestingly, nobody gets sterile immunity. If you go to a village, and this has been done, if you bleed everyone, say, every day for a week, you'll find that everyone at some stage has parasites in the blood. Quite different from the immunity that you might get to measles or mumps or smallpox. Something else is happening. And that persistent infection is required to maintain immunity. So just to summarise this point that we need to think about, which is going to be really important when we come to the picture that Mark asked me to talk about, the global approach to eliminating malaria, these points have to be taken into account. The best way at the moment, in the absence of a vaccine, to make you immune is to have multiple episodes as a child, to get through those with appropriate treatment, and then give them a clinical immunity that protects them for the rest of their life. If they could get to that stage without suffering these consequences, they'd be a position that would be very good to do. This is a really big problem. Immunity is lost if exposure is reduced by a program, even a vaccine that induced sterilising immunity for a couple of years. When that wore off, or if you put kids under bed nets for a couple of years, they're then at increased risk of malaria when they come out of that period, and potentially at risk once again. There's an exceptional thing that happens in pregnancy, and particularly the first pregnancy, when they become susceptible once again. And the reason that they're susceptible is that within the placenta, this is placenta and this is the maternal side here, this is the baby's side here, and the malaria-infected cells here line up along the placenta and interfere with placental function. It was Stephen Rogerson who discovered the molecule that leads to this interaction, and it's a subject of great interest to see if you could interfere with that interaction to prevent malaria in pregnancy. So girls are completely susceptible, like the boys are resistant, then at their first pregnancy they suffer this problem. And it appears those antigens on the surface of the infected cells that I referred to that can vary, if they get a variant that can stick here, that leads to the pathology. So it's probably these antigens on the surface of the malaria-infected cells that are responsible for the organ-specific pathology, potentially say in the brain or the placenta or in the lung, as examples of how this may happen. Now, if we start now getting more to the public health aspects of malaria, some understanding of the disease was important in order to understand the global efforts to control it. Quite different from, say, a global effort to control polio, where it's quite clear it's person-to-person spread, there's a virus with 3 different antigenic types, get everyone immunised, it'll work. Similar with measles, much harder for malaria. So what happens in that, back to the host parasite and the environment, this is a picture from Indonesia, from the western Papua, in which we see they've had transmigration problems from Java into Papua. And where they've opened up the forest, what's happened with non-immune migrants, that people living here are non-immune and therefore exposed to malaria. And what happens there, rather than people being infected at night when they're sleeping, it happens during the daytime in the forest. Now, this will become important later when I explain about our control methods. So the mosquitoes bite outdoors during the day, and malaria occurring in all ages. The relevance of that, perhaps I could explain now. One of the main methods for reducing malaria is to spray the walls of houses, and that relies on the fact that most mosquitoes that carry malaria bite indoors during the evening and then go and rest on the nearest available surface. There, if the DDT or something's on the wall, they'll ingest that and they will die. Now, here's the problem. If the mosquitoes are a totally different behaviour pattern, how do you tackle mosquitoes that are biting during the day? And that's a huge challenge, and particularly these mosquitoes that carry Plasmodium vivax. So what in fact does cause malaria? It's too simplistic to say, oh, well, it's a malaria parasite in a mosquito, because look at the factors. Climate, and for the life of the mosquitoes, it will depend on rainfall, altitude, temperature, for those cycles that I've already described. So the breeding sites and the habitat. So that, for example, the mosquitoes that transmit dengue tend to bite round your house in small pools of water, and you can get rid of them pretty easily. Much harder to tackle the breeding sites for malaria, which are more broad spread. Very, very difficult indeed. The housing, are they close to the animals? Are they screened? Personal protection, and then the characteristics of the host. So any one of these factors altering the balance can lead to an increase in malaria. So let's say some of the genetic things which are extraordinarily interesting. West Africans are totally immune to Plasmodium vivax. When those malaria parasites burst out of one red cell and go into another, The parasite needs a receptor interaction at the surface of the red blood cell. And you've heard of blood groups A, B, O, AB, etc. Well, there are minor blood groups, the so-called Duffy blood groups, and the Vivax molecule needs to recognise that receptor to get in. And West African blacks simply do not have that receptor, so they're 100% immune to Plasmodium vivax. That's incredibly interesting that that's happened. There must have been a fluke event, and then over a very long time, that's led to a population with that huge survival advantage. In fact, many of the abnormalities of red cells and haemoglobin provide some protection against malaria. So you may have heard of sickle cell anemia, which is an abnormality, hemoglobin S, or thalassemia. Both give a degree of protection against malaria, and they particularly protect against Plasmodium falciparum. So, of course, the survival advantage there, in the case of sickle and thalassemia, is balanced by the very negative effects on homozygous individuals who suffer terrible consequences, so it becomes a balanced polymorphism. So it's very interesting for those who are the evolutionary biologists. It's widely thought, as I've said before, Vivax can survive in quite remote communities, and probably was able to survive in hunter-gatherer communities with that ability to survive a long time without requiring constant transmission. And possibly falciparum really only came when people start to live in groups, in villages and so on, from being hunter-gatherers. Interesting discussion, no time for it here. So, and obviously requirement for mosquito transmission defines susceptible regions that may change with season or with global warming. So that in Papua New Guinea, for example, there are places that can have epidemic malaria kind of in the highlands where there's a balance whether it can or can't from year to year transmit. In the Sahel, as the Sahel coming south with drying of the desert and desertification, some places that had malaria, there's becoming less. It's not certain what would happen with global warming, but in some places there'd obviously be more and some places less. But suffice to say that there would be change. So I've talked about some of the genetic things, and there's also some innate factors that we don't really understand. So that in some model systems, people used to try to do vaccines. Malaria in birds is actually quite common, and there's models that were done for vaccines where in ducks, for example, because they're easy to keep and manage, but you had to do your experiments in ducklings because within a matter of 8 or 10 or 12 weeks, a duck is totally resistant to that malaria. And so there are huge age-specific effects as well, and it depends on the model. There are also huge non-specific protection. There are animal models in mice where you can protect mice from mouse malaria with BCG or other adjuvants, Corynebacterium parvum. In other words, non-specific protection, not well understood. I've talked of acquired resistance and the fact that we practically never had sterile immunity, that so often we get this phenomenon. And of course, when you're out there trying to study this disease, you can't say who is or is not immune. All you can say is that if someone gets malaria, that they're not immune. And even someone who's sick, in fact, could be sick from another cause, but find parasites in the blood. So it's sometimes hard to say to people they have or have not got malaria, which makes it challenging for us. So I think it's really important to understand this concept of concomitant immunity because it's going to help us. Now I think it's interesting for the immunologists in the room I've referred to things like measles or polio, where it's an infection cure and you're cured for life. But I really think it's quite interesting to see there are other diseases where we've got similar types of things that you'd like the immunologists to start thinking about. So I've said the issue about malaria being more than one disease. HIV is the same. Certain strains, for example, give cerebral manifestations and dementia and so on that we don't fully understand terribly well. The other, I've talked of concomitant immunity. But it's really interesting, if you got infected with HIV today, on average it would be 8 or 10 years until you started to get AIDS, which means that at some stage in the course of your illness, you're controlling the infection, and of course some people are long-term controllers, And what are the mechanisms here that are controlling infection that we should be trying to understand? Many variants here, and of course we've talked of the Sickle and Duffy groups that protect from malaria, and there are similar things in HIV as well. And what's interesting when you're discussing approaching these sorts of things, so say in the malaria case, and the same thing applies to HIV, should you be trying to mimic this condition, or should you be trying to produce sterile immunity? Huge debate. Similarly, with the huge number of antigenic variants, you'd say, well, antigenic variation is important, therefore they're clearly important to immunity in a natural setting, therefore that's what we should be targeting. But then, is that right or wrong? Others would say that's completely wrong because the parasite has got the ability to change so quickly. And what you should try to do is to find an antigen that's not normally immunogenic and then make it immunogenic. Now, for an infectious disease physician, that's pretty interesting as well. Traditionally, until a few years ago, there were very, very few examples where an immunisation was better than natural infection and cure. One of the very few examples was tetanus. If you have clinical tetanus, you can still get tetanus again, unless you immunise. So immunisation against with tetoxide is better than infection. But for most infections, very unusual until the work with HPV, which showed if you dress up an antigen as a viral particle and with the right adjuvants, you can do better than nature. And similarly, with the first vaccine against malaria, is a kind of novel antigen and made immunogenic in a way that you don't see in the field. So possibly an approach should be to find something that's not normally immunogenic and make it so, which is quite interesting. So that's what I say, that adjuvants and delivery systems, particularly in the vaccine field, as delivering with viral particles, has been a new way. Many people discuss whether animal models are useful, and in this matter we say, what's more interesting, human malaria in a monkey, or mouse malaria in a mouse, or monkey malaria in a monkey? The debates go on forever. So I think the comparison with the other disease, we've got quite a lot to learn. So we've said that the comparisons, there's a lot that can lead to. So for example, it's sort of opposite. When HIV infection, you get a waning immune control leads to disease. So you start kind of immune and then you're susceptible, whereas malaria, it happens in an opposite direction. So I'll just move on a little bit. I'm a bit concerned about the time. We've got, therefore, the world situation at the moment, the annual death rate of about 700,000 people with 500 million people infected, high percentage of the world at risk, particularly, of course, when you put India, China and Africa, and huge economic loss. And interestingly, it was the huge economic loss that made people 10 years or so ago say more should happen. And so the distribution of malaria is where you would expect it, with the distribution of the mosquitoes around the tropical region. But it is declining. If you look at the world map here in 1948, tremendous number of countries involved, including Northern Australia. And in the 50s, there was a big program to try to eliminate malaria, particularly with the use of spraying. For the reason I've said, mosquitoes feed, rest on the wall, and then they die. So if you can do that twice a year, that should be enough to stop the transmission of malaria. And it was very, very successful indeed, and worked very, very well in many countries, and eliminated the malaria from southern US, southern Europe, and some other countries. So the map shrunk considerably by that stage. But with respect to the global campaign to eradicate malaria, certain lessons were learned. And this is a study from Sri Lanka. 1946, DDT spray came in, very successful. 10 years later, they'd fallen from 3 billion cases a year to 7,000. So they thought the job was done. In the mid-60s, malaria was solved. What happened? Within 5 years, resurgent in cases, 500,000 cases. This is a very strong lesson now for people who are arguing for control or elimination, because from what I've said before, you'd be right if you'd guessed that when it resurged, it wasn't just in children, it was in people of all ages. 5 years without exposure to malaria, people were non-immune, and people of all ages were at risk, and the gains were all lost. So why did it come back? Well, first of all, they said we don't need the programs because we don't have malaria. There was opposition to the use of chemicals such as DDT, but it was really using them from agriculture and spraying them over huge areas that was more of a problem than individual house spraying. Drug resistance was a problem, and some insecticide resistance, and people had lots of reasons why it came back. So what's available now? There is the methods for prevention, which is spraying houses and also bed nets, medication in target groups, better management of cases, but most importantly, we need a surveillance system that can respond. And the problem is that what you require for this to happen is to have a strong health system, such that cases can be rapidly diagnosed, reported, and go back to them. So there's an outbreak in a village in Papua New Guinea, quickly get in there with all your control measures to prevent it. And in the countries where malaria is a problem, health systems are obviously weak. How do we tackle these areas? As I've mentioned before, these are not susceptible to the usual things like indoor residual spraying of houses, and it's malaria in people of all ages. So these are the tools that we have, and as you'll notice, there's really nothing more than we've had in the past. What's happening here? We seem to be going backwards. I don't know what's going on. I think I've mentioned these before, the problems. The alternative are bed nets, which have been very, very effective, not just bed nets themselves, but when impregnated with insecticides. If you give them to every pregnant mother, you'll have very favourable effects on both the children and the mother's health. For people living and working in the forest, using hammocks and spraying with insecticide can have some of those same effects. Big challenges with the nets. They're public health things. The people in basic science often are not aware of the challenges. You think there's a solution there, but it's not available. So the nets need careful treatment. Their variable efficacy, the problem of who pays for them. And there's a lot of thought now about how we make these things sustainable interventions, because what we need to do is get people out of poverty. And one very good example from East Africa was, instead of purchasing them from overseas, they gave people vouchers, every pregnant woman a voucher. She goes to the store to buy a net, and that stimulated an industry, local industry, to build the nets. And this is what we really need to do, sustainability for the future. Diagnostic tests are a huge problem, as I've said, because of microscopy. The good thing now is there's an antigen detection test for Plasmodium falciparum, but the ones that we need for Vivax, this alternative form, is not yet there. So the other problem from a public health perspective is, at the time when malaria is getting very low and you're trying to eliminate it, the vast majority of your tests will be negative. It's pretty depressing to do 100,000 microscope slides and not find one that's positive. So why do you keep it going? And not only that, if you, in a country where you're trying to eliminate it from this side, there's no malaria, whereas over here there's lots of malaria, people say, why are you spending all your money where there's no malaria? It's very hard to understand that argument. And similarly at the moment in the global program against polio, huge amounts of money are being spent for a matter of a few hundred cases per year for the same reason, and it's criticised for the same reason. So to make it work, you need a very good functioning health system that can provide all the aspects you need. It's no good if the bed nets are sitting on the wharf and not delivered, so you need good ways of getting the medicine and technology there. You need service delivery and above all training the people to deliver on that. For those not in public health, this is an obvious slide, but perhaps it's not to everyone else, but it's really important. Let's say you have this is a real example of a combination therapy, which we recommend now to prevent emergence of resistance. Let's say the combination, if you gave it to people while you watched them and supervised treatment, was 98% effective. Let's say that could only get to about 60% of the people who needed it. The diagnostic test was 95% accurate. The provider gave the right treatment 95% of the time. The patient took it 70% of the time. The final result of effectiveness is 37%. So a very, very effective intervention or drug, when you start to apply it at community level, less than 40%. This is a really important concept in public health, like, you know, that condoms prevent pregnancy or condoms prevent HIV, but if you don't have them accessible, available, used, etc., you won't achieve the public health outcome that you desire. And this is particularly true for malaria, and as we drove for eradication, say for bed nets, if there are not many mosquitoes and not many people are getting malaria, what's the incentive to keep it up? Now in 2007, Bill and Melinda Gates said, look, the world's sitting back too complacent, we should be thinking about eradicating malaria. It's intolerable to allow the burden to continue. Some people were shocked and horrified and said, did they really say it? They should never have said it. Irresponsible, terrible. But what they said was that, why not try and eliminate malaria? Elimination we usually mean from an area, like elimination from a country, like Vanuatu, Solomons or Burma. Eradication is to eradicate it from the whole world. And the question is, can it be done? The answer is, with currently available tools, well, the answer is lots of countries have done it with what's available now. Some would argue that in Africa, where intensity is so intense, the transmission, that it can't be done with what we've got. Political context, it requires money and political will. With current health systems uncertain. Undoubtedly, enormous amounts can be done. Can we get the whole way? It's hard to be sure. So, I think there's general consensus in the scientific community that it must be the long-term goal, and particularly we need to set the research agenda now. And when you start to think, from what I've given a lot of background about the difference between infection and disease, if you think about control, you're reducing illness and death. If you think about eradication, you've got to block transmission. Similarly, if you're getting you've got to think of not just the one that's the main killer, but the other one that causes anemia and less severe disease. So this is really important changes, and this is what's led to a new research strategy. And the goal being to reduce R0 to less than 1, to block transmission. Transmission of malaria is very hard to measure. It's all right when there's a lot of malaria or practically none, but very hard in the lower reaches. The global partnership to do this, Roll Back Malaria, brings together lots of people to try and speak with a unified voice. And anyone who's a researcher is part of this, research and academic. I happen to be the representative on the board at the moment, so we do get a voice at this. Of course, we're always arguing for more research, as you might imagine. Multilaterals, WHO, World Bank, UNDP, and these are the people that put together the money, the budgets, and the challenges to take it forward with the country, like the Global Fund for AIDS, TB and Malaria. And I think Bill and Melinda Gates deserve tremendous credit for kicking this off. So what you do is start with increasing the control to get a bigger impact, get it for a while, then when you have about less than 1 case per 1,000 people per year, you can think about the challenge of elimination. And this hasn't translated perfectly, but in our region, there are countries like Papua New Guinea and Laos a long way away from that. Then you have countries that are getting closer, and this is China, Vietnam, Korea are here and aiming for elimination. So it's really important that these countries go the full way to get rid of malaria completely. But then challenges arise. Now this is some work that's been done in Africa, but also repeated in the Pacific. So they examined the malaria, the Anopheles mosquitoes, before and after a program for insecticide-treated nets. This was in Tanzania. And what they found is that before and after this program, an increased proportion of mosquitoes were feeding outdoors after the program. In other words, the population was dominated or was being increasingly outdoor biting mosquitoes who could avoid the nets. And so there's chemical and biological resistance that's being looked at. Not nearly enough research is being done, and so it's a big challenge to deal with it. Plasmodium vivax, as I've mentioned enough, it's not so many deaths, but it's a really big problem. And this relapse is a real problem because there's a reservoir of people living in the area who are going to start new epidemics. And the drugs that are used on the liver stages are unfortunately potentially toxic and can't just be given under mass administration. In the research agenda that we set, finding a new drug for these liver stages was very high on the list. And this problem of outdoor biting. So it's a big problem, and that's one to be resolved. Another really big problem is the resistance to anti-malarials. The latest and best is artemisinin, and now the world suggests it should always be used in combination, but the time to clearance of infection is getting longer and longer. It was, when it first came in, infection will be cleared within a couple of days. Now in lots of these hotspots, it's been seen that it's taking longer and longer, and it's highly likely that higher grades of resistance will occur, and there's no obvious successor to this drug to be used on a wide scale. Very interesting things that get thrown up from time to time. In Malaysia, an astute parasitologist looked down the microscope and saw some funny-looking parasites. And these parasites, for the experts, looked a little bit like falciparum, but not quite right, and there were funny forms they didn't quite understand. So they did some genetic typing and found that this was a parasite that occurs in monkeys, Plasmodium malsii. And so this is the monkey that carries that parasite. It had been known from a laboratory accident years ago that it could affect people, but here was an example that it had turned up again. Fortunately, it was susceptible to normal drugs. But then what happened, having first been reported in Malaysia, they then found it popping up in lots of different places. And once again on this idea of the host, the microbe and the environment, what was really interesting was that this parasite occurred where this parasite group was detected in all these places. And what was happening with loss of habitat, it used to be that those mosquitoes were only up in the canopy with the monkeys where malaria was transmitted. But as forest was lost and people were living close to the forest, then people became infected from the mosquitoes that had also fed on the monkeys. A really good example of the sort of thing I started with at the beginning. Malaria also pops up in different places with all the travel, and I think that it's really going to be an increasing problem with people moving around the world. Even in the north of Australia, there can be susceptibility. So we're going to see more of malaria. It's alive and well, even though overall there's such a promising response with the global effort that's being made. So those are just some examples that came from Australia. And to remind us all that malaria is still with us. I think that there's some, I've tried to give a balanced picture. There's been tremendous progress on the control of malaria over the past 10 years. We always used to quote 1 to 2 million deaths. It's clearly now a lot less than that. Bill and Melinda Gates have brought the world's attention to malaria and shown that with application of known methods we can make a huge effect. But I think as scientists, the problem is that people who are not scientists have taken a very simplistic approach to what's happening. There's overall optimism, but we know there's going to be drug resistance, insecticide resistance, and we just must maintain the course. It's rather good that the Australian Aid Program is doing quite a bit in the Pacific and wants to continue to do more. But as I've said, what we really need is a game changer. And has anyone here talked about Wolbachia and dengue?
Mentioned it.
Yeah. Well, just to remind for those that haven't, there's a commensal organism that can occur in parasites, but also in mosquitoes, called Wolbachia. And sort of 20 years ago, it was a pain in the neck for people trying to grow mosquitoes because they find this funny organism in the mosquitoes. They still survive. But then, by good luck, some people used these mosquitoes in their experiments to transmit dengue. And what they found was that the mosquitoes became resistant to dengue. And not only that, the mosquitoes have a survival advantage to the extent that these mosquitoes can be put into community, and then they will not only stop dengue in themselves, but that will populate what we'd like to do. And the other, and this is work that's been Scott O'Neill, Larry Hoffman and Cam Simmons, who's just come back from Vienna. Fantastic work and really interesting. Now, as you could imagine, people are now working very hard to see if that could be transmitted into the anopheles, the vectors of malaria. And this is the type of thing, the game changer, that could potentially give a new weapon that might work in heartland Africa, where there's intense transmission. And this might be enough to just make it a bit of an easier target when combining all these factors. So I hope I've been able to give a bit of an overview of host parasite and the environment and what's happening against malaria control and potential for elimination in the years ahead. Thanks, mate. Yeah.
Questions for Graham, please. I'll start. There seems to be a real major research block in certain infectious diseases, and you mentioned one of them, which strikes me as being quite funny, is that we have, apart from chloroquine, we have no drugs against the hypnozoites. And to me, it looks very superficially as if there's very little research in that quarter. And you can also say that of tuberculosis, where it's been 3 decades now since we've had no anti-TB drugs, yet there's such major diseases. What are the research blocks? Are people not interested in doing the research? Are people not funding the research?
Well, when we had the malaria eradication research agenda, that was the top priority. We can't even culture Plasmodium vivax in the laboratory, and there's not a satisfactory animal model. And so when people want to do challenge experiments with vivax, they have to take blood from people to infect the mosquitoes, to then challenge other people with all the hazards that might involve. We argued that we need to put in place the basic research for, first of all, growing liver cells, sufficient capacity that they can be cultured. And some of you might be aware of the work that Louis Schofield's been doing, because the Plasmodium vivax grows in reticulocytes, the immature red blood cells, and so it's very hard to get a supply of those, and it hasn't been cracked. It's just terrible. And also, like the dormant forms of tuberculosis in the lung, I don't think there is a model for them, is there, as to these dormant forms that you have after you've been infected with TB. It's the same thing. Very hard, very long term. In 1955, it was said that the prime need for malaria control and elimination is a drug against the liver stages. Still hasn't happened. So it's a real worry.
Ben.
Hey, Graham. So the idea was always it takes a long time to acquire immunity, that it was just that the epitopes that were protective were hidden, and it took a lot of bites for your immune system to spot those. But that doesn't explain why immunity wanes. So is there any notion of what happens there?
Well, that's just a huge challenge. I mean, the vaccine manufacturers for HIV malaria are all very interested in long, P-dependent memory, or what is it? We simply don't know. And the problem is that right back from when we always studied, you can't tell which person is immune or not immune because you don't know what to study. But, in other vaccines, for example, now we're seeing whooping cough in Australia for waning immunity. That's kind of decades. Whereas here's something that goes after a few years. And no one really knows. We need immunologists to be working on malaria, I think. So, or more immunologists to be working on malaria to understand these things. But my understanding is that, in this very room, or in the old Wehi, discussing what is memory, do you need persisting antigen to have long-term memory? And I understand that it's now recently been proven over time, telling me that no, you can have long-term memory without persistent antigen. So that was always potentially thought, well, if you don't have constant antigen, you can't constantly boost. But that doesn't really make sense. And the other thing, interesting dilemmas. For example, we all know that with HIV, and when you get AIDS, there's a huge increased risk of getting tuberculosis. There's a huge increase of bacterial infections from pneumococcal disease. There's a bit more malaria, but not an awful lot. So it would suggest that we don't really know babies who are born with HIV, whether it's harder for them to become immune to malaria than kids who don't have HIV, but the maintenance of immunity to malaria does not seem to be heavily infected by the presence of HIV, whereas it clearly is in TB. There could be a clue there. They're different diseases and different, presumably there are multiple mechanisms of memory.
So you said it was known previously that Knowles eye could infect humans from a lab accident. Is there any such knowledge about other strains that are currently only found in animals that would give an idea of the zoonotic potential? Say we managed to eliminate all current human strains, what would be the risk that, you know, a few decades down the line, there'd be a whole new species of malaria to contend with?
Well, I think that's a really important question, because of diseases that you're trying to eradicate, you want to know there's no animal reservoir. Now, there's a Plasmodium innui, which is a malaria of monkeys in the Amazon, is thought to be very close to Plasmodium malariae, which was the fourth one that I didn't even mention today. But as far as we know, the malaria that infect penguins or lizards or birds usually don't infect humans. So possible, but there would always be the threat of Knowles eye, but it's relatively confined. And so, but that's an example that would have to be got rid of. Let's say that became drug resistant or something, that would be a real problem.
Graham, are there any discussions around, well, there are discussions around light attenuated vaccines? What's the stomach for that, and obviously the inherent risks that are associated with it?
Yeah, well, that's a really, really interesting question that we could have spent the whole time on vaccines, because it is just so interesting. It's been known, just the background for the non-malaria people, it's been known for 40 years that if you take infected mosquitoes that contain the infectious form of malaria and you irradiate them to attenuate, and then feed them on people and you need multiple feeds of many infected mosquitoes, those people get sterile immunity for malaria. And it's just amazing, and that's been known for a very long time. Now, for a long time, people said, okay, well, let's study those people to see what's the immune response that protects them, and then maybe induce that immune response with a single antigen. That was one approach. Secondly, another approach was to say, well, now that techniques are coming in, could you make a vaccine in this way? Now, people initially thought to have a commercial vaccine that depends on manufacture of literally millions and billions of sporozoites would never work. But someone's actually attempting that right now. But a better and a smarter way would be to have an attenuated sporozoite vaccine, which means you have something like other vaccines that it's incapacitated, so it lives long enough in the liver to stimulate immunity and to protect you. I think people still think, no, that that's a very viable option, and I think they should press on hard. There's always the problem that it could revert, and as you know, there have been some experiments. But it's also happened in bacteriology, where people thought they'd put a lethal mutant in an E. coli or something, then the pathway's been bypassed. So I think that has clearly got potential. Now, the other form would be that some people say that the only way is think of those children with blood stage infections. Other people said, why not create a vaccine that's an attenuated blood stage vaccine? Now, to do that, of course, the thought of injecting blood into other people is always a worry. Now, some experiments in that form have been done, where what they did was to infect two people and then tested them for everything they could think of, and infected two people and froze down thousands of aliquots of their blood in vials. Then they waited for 3 years to make sure that people didn't get hepatitis or HIV or anything that you can measure for. And those have been used in a series of experiments, and they've been extraordinarily interesting, because they've shown that after a very small number of infections, people appear to become immune. So it could be something to do with the dose phenomenon, that a low trickle dose could be what was required to make you protected. Incredibly interesting. And we also know that from other forms in parasitology, a model system work that Emanuela Hammond and Graham Mitchell did a long time ago, will tell you that the immune response that you get is different if you give a small or a large dose. In models of the disease leishmania, in mice, you can give a high dose or a low dose and get a totally different readout. One leads to lots of IgG and no protection. One leads to a T cell response that protects. In fact, that's how the Th1, Th2 was just sort of discovered and worked through in that model. And it could be this low-dose model of recurrent blood stage infection, to everyone's surprise, gave great protection. Now, that work's going on, but it's highly controversial as to whether it would ever lead to an attenuated vaccine. But in principle, you would think that an attenuated vaccine is the most likely way of achieving the desired result.
Can you please join Graham outside if you have any more questions? He'd love to sort of answer them for you. And I'll thank you very much again for a wonderful lecture.